This is part 1 of 3 of the Poinsettia Diagnostic webinar offered by e-GRO.org. In part 1, Kelly Ivers from North Carolina State University will cover common poinsettia diseases. In the following parts, part 2, Brian Whipker from North Carolina State University will cover common nutrient disorders of poinsettias, and Raymond Cloyd will cover insects and mites in part 3 of the webinar.
I'm trying to forward my slide, but it's not letting me. Can you forward my slide? All right, so I just wanted to mention before we get into talking about poinsettia diseases, I wanted to mention that there are a number of resources that are available at this website. It's the North Carolina State University Ornamental Plant Pathology website. And on this website, we have a lot of information. It's not just on boxwood blight, but also with commercial nursery and floriculture information. And it includes chemical control recommendations, as well as some tables on different sanitizers that can be used in greenhouses. Next slide. So this slide actually is a data slide that Colleen Warfield had put together, and I thought, even though it's older data, it shows really good information. And what I mean by that is that it makes a good point. Basically, you can see the months below: August, September, October, and November. And these were actually diagnoses. These were samples that were sent into the Plant Disease and Insect Clinic from 2002 to 2005. And so on the left, we list a bunch of different poinsettia diseases and when they occurred throughout the different years. Now, it shows a few things. What it does show is that Pythium is problematic throughout the entire production season of poinsettia. However, there are some diseases that are just associated at the beginning stages when they're cuttings, and this would be scab and Erwinia. It also shows that you can find Pythium most years in greenhouses, and as well, it shows that some diseases are actually more problematic in different years. This might actually have to do with the source of the cuttings and etc. So next slide, please. So the way that we're going to talk about poinsettia diseases in this part of the webinar, and I think throughout the entire webinar, is to focus on which part of the poinsettia it attacks, or that you're seeing symptoms. And this will give you a good idea as to how to do some of your own informal diagnoses. Next slide. So the first set of diseases that we're going to talk about are ones that attack the roots. So let's go ahead and get started with that next. So just to kind of give you an idea, poinsettias are very susceptible to Pythium root rot. And I'm going to focus on this pathosystem for most of the portion of my talk, because I feel like it's the #1 production issue that we see throughout the entire Southeast, and most likely throughout the entire US. So this disease often starts during rooting of the cuttings, and that's why we tend to see this disease throughout the entire poinsettia season. But it can also be problematic in ebb and flood systems, and so you can see it also occur later on in the production season as well. Pythium species they're typically soil-borne, and they spread via irrigation water, and we'll talk about that in a second. But they can also spread through the use of contaminated media, pots, and rooted cuttings. And it really seems like Pythium has become very well adapted to poinsettias, because we see this being an issue in many greenhouses. Now, what's interesting about Pythium is that it tends to be a root nibbler. On other crops in greenhouses, we see that it attacks the roots, but in poinsettia, it can also pretty much attack the entire root system. So with Pythium on poinsettia, you tend to see a very poorly developed root system. However, if the plant gets infected later on, you will see more dead root systems throughout the pot. Forward, please. I'm going to see if maybe I can't forward it, because I see an arrow. So I'm going to, I'll try to do it the next one. So with Pythium root rot, the roots appear water soaked and discolored. Let's see if I can, no, it's still not letting me, it's still not giving me, so can you go back one? Yeah, there we go. So with Pythium root rot, the roots appear water soaked and as well as discolored, and it can cause root sloughing as well. And what I mean by root sloughing, forward please, is that the cortex, the outer cortex, the barky part of the root will pull off. It pulls off very easily. So the only thing that you can really confuse Pythium with, I guess Phytophthora can be an issue, but in the cutting phase, we also see Erwinia. But the difference is with Erwinia, which causes it causes a soft rot. Erwinia is easily distinguished since cuttings can fall apart and completely collapse. And usually this occurs in a very slimy mass, and it's got a very fishy odor with Erwinia. So Pythium root rot is different. You tend to see brown discolored roots as well as root sloughing. Next. So often a Pythium can appear later on in the production season, and this can have a big impact on plants as they get further into the season because it's very costly. So it occurs when plants are closer to finishing. And often it's very common that the plant will wilt, and sometimes you'll only see one stem or one cutting in the pot that will wilt in a pot that has multiple cuttings. And so often growers and production managers will wind up watering this plant a second time or even more often. And this is really the last thing that the poinsettia needs because the root systems are rotting and they're breaking down. And what I have to remind you of is that Pythium really likes water. So when you tend to overwater and when you water infected plants, it can be spread in these ebb and flood systems. Next, please. I just wanted to show you a few pictures of what Pythium looks like. And again, you can see here, this is just the start of Pythium. And you can see the one plant a little bit off to the center where a portion of it is starting to wilt. This is a very common symptomology of Pythium root rot. Next. So I do like to talk about Pythium in general, and so the next couple of slides I'm going to talk about some research that we've conducted at North Carolina State University to talk a little bit about Pythium so that we can understand more about its role in greenhouse crops. And I feel like this is important because often more than poinsettias are grown in a production area, and what you need to realize is that those other plants can also be potential hosts. But at our clinic, and it's throughout a lot of clinics, we tend to find Pythium very commonly. So Pythium is the most commonly diagnosed disorder of greenhouse crops in North Carolina. However, most clinics, they don't identify the species. They don't test for chemical sensitivity, and what I mean by that is their sensitivity towards fungicides, and we really can't offer an insight whether Pythium is isolated, the Pythium that we isolate from a sample, whether it really poses a production problem. And that's because we have many Pythium species that are very common secondaries. So when you find a Pythium infection, unless it's identified to species, it's very difficult to say whether or not it's pathogenic. Next, please. So I wanted to go over a little bit about our recommendations and how we recommend the control of Pythium. In the past, and it was used pretty much overly used throughout the greenhouse industry, and not just for Pythium, but also for Phytophthora, is Mefenoxam, or the older chemistry was called metalaxyl. This is in a fungicide mode of action group that we call FRAC4. However, another chemistry is very commonly used. It's called etradiazole, and it's the active ingredient that's found in Terrazole or Truban, and it's also one of the actives that is found in Banrot that is commonly used. However, there are a number of other chemistries that we find can be effective on poinsettias to control Pythium. And one of the ones that we've been really interested in North Carolina is a fairly newer product to greenhouse production. It's the active ingredient is called cyazophamid, and we know it as Segway. This is produced by FMC. Adorn can also be used. So Pythium is very closely related to Phytophthora. So we tend to see a number of the chemistries that are used for Phytophthora also used on Pythium. However, they don't always respond the same. We tend to like etradiazole, and cyazophamid is also a product that we like to recommend for as a rotation product for Pythium infection in poinsettia. Next, please.
Kelly, I think I have it figured out if you could go ahead and try to advance it yourself.
Okay. Yep. Was that me?
It was my problem.
Okay. So, all right. I get to forward. I'm excited. I do want to mention, though, a little bit about Mefenoxam resistance. This is becoming more predominant throughout the greenhouse industry in the United States. And it's because Mefenoxam was overly used throughout the entire ornamental industry because it was so effective. However, Mefenoxam resistance to Phytophthora and Pythium is very widely documented. And there's been a number of publications that have come out recently. There was a publication in 2013 by a postdoc and actually a graduate student at NC State University. Her name was Heather Olson, and she also documented widespread resistance of Mefenoxam in Phytophthora in floriculture crops. And Gary Moorman at Pennsylvania has also documented this, as well as research in California. And so, although it might not be that there's complete resistance to Mefenoxam, we do like to talk about using rotation chemistries, and this is where Emma Lukabaugh's research is really interesting, and it's fairly recent. It was conducted in the last couple of years, and so I'm excited to share it. So one of the things that that we were curious about when Emma was conducting her research is what species were predominant in North Carolina and in floriculture. And so one of the questions was, well, why do we need to know what Pythium species are common in greenhouses in North Carolina? And the reason for that is because there's over 200 different species of Pythium. And like I said, many of them are common secondaries. There's not too many that are highly pathogenic, and there's really only a few that are pathogenic on poinsettia that we'll show. So species really differ in their aggressiveness on certain hosts. And so we're going to look at that. But also, many different Pythium species differ in their sensitivity to fungicides. And what we're finding is that there's local selection pressure. And what I mean by that is different greenhouses will build up selection pressure to Mefenoxam and other chemistries, and it correlates back to their use of these products. And so some species of Pythium have found never to be sensitive to Mefenoxam. However, a number of species have always been sensitive. And so really, the more you know, the better the diagnosis. So we really feel like identification down to species could be very helpful to the grower, because it can help you with, and as well as diagnosticians, better formulate control recommendations. So I'm not seeing that arrow down there, so it's not letting me forward. Oh, uh-oh. No, it's okay. All right, so I'm going to talk a little bit about Emma's project. Again, she conducted this in the last couple of years, and so what she wanted to do, her major advisor was Barbara Shue, who's the director at the Plant Disease and Insect Clinic, and Emma has worked there for a number of years. We went ahead and collected samples of not just poinsettia, but a number of other bedding plants throughout the entire state of North Carolina. So we had 26 greenhouses that we looked at in 21 different counties, and Emma collected over 360 Pythium isolations from these 26 greenhouses, and this spanned 36 different host species. This is not letting me forward. So again, I know this webinar is on poinsettia, but we're going to go ahead and just talk about some of the common greenhouse hosts to Pythium. 21% of Emma's isolates came from poinsettia, and secondarily, a number of them came from mum, followed by snapdragon, petunia, geranium, gerbera, lantana, fuchsia, vinca, and verbena. And then we did find Pythium on some other crops, but it really represents a very small percentage of her isolates. Forward, please. So we've always heard that people have done Pythium surveys and tried to determine what Pythium species were common in greenhouses. And these results were kind of interesting because I've always seen the list include Pythium ultimum. However, if you look at the top 3 species that we found in North Carolina that spanned all those crops that I mentioned, Pythium irregulare and Pythium aphanidermatum were the most common ones, followed by Pythium myriotilum. So it was interesting. We tended not to see too much Pythium ultimum, as well as a number of other Pythium species. And again, Emma's work really focused on morphology as well as DNA-based diagnostics. So it could be possible that we're finding some new things based on the way we chose to conduct our survey, but also on our methods. Forward. Okay, you can forward again. So I want you to focus to the very right and on, oops, back please. So I want you to focus to the very right, and you can see on the lower axis on the bottom, you can see a list of all of the hosts that we sampled from. Oops, can you go? Okay. So again, I want you to focus on the very right and look across the different crops that we sampled in North Carolina and look at the species diversity that we found. And since this is a poinsettia webinar, we're going to focus just on the poinsettia column. So you can see that there were 4 species that were found, but predominantly we found Pythium aphanidermatum as well as a small number of Pythium myriotilum. That were associated with poinsettia. So 91% of the isolates that we collected from poinsettia that showed Pythium symptoms, they were Pythium aphanidermatum. So this is going to be the main player, and this is the one that I'm going to focus on when we continue to talk about poinsettia and Pythium. Forward. One thing that I like to mention is that Pythium aphanidermatum, it's been known to be Mefenoxam problematic, but it's also been known to produce an abundance of zoospores. Now, they use the word zoospores in the spore stage because it is motile, which means it has the ability to move or swim. It's got a flagella on it that actually allows it to move in aquatic systems. So while we know that Pythium is a soil-borne disease, we also can tell that it has an aquatic stage. And because of this, it's especially problems in ebb and flow systems. Next slide, please. So with part of Emma's research, we wanted to look to see if we could develop a Mefenoxam sensitivity test that we could use in the clinic to offer as a service. And so one of the things that she did is she validated a protocol in order to do that. And she used this protocol across the range of her samples to see if there was a mixed sensitivity with Mefenoxam across these crops. Now, this is going to be dependent mainly on the species diversity that you find within a crop. For instance, you can look at gerbera and you can see that all of them were resistant. And that's because it has to do with a particular Pythium species that was found that were predominant in gerbera. However, with poinsettia, again, I mentioned that Pythium aphanidermatum represent 91% of the actual test that we conducted with Mefenoxam sensitivity. You can see that we had a mixed reaction. So some isolates were resistant and some were sensitive. So when you think about it, I still feel like Mefenoxam can be a valuable tool, but what it also shows is that you cannot rely on this product as a single mode of action throughout your entire season, because you'll wind up knocking back the sensitive isolates and you will develop an entirely resistant population. So if you don't rotate, you'll wind up losing a lot of the efficacy of this particular active ingredient. Forward. So just a little bit to highlight some of the things that we found. There was a strong association of Pythium aphanidermatum with poinsettia. And these Pythium aphanidermatum populations did have a mixed sensitivity to mefenoxam. Now, Mormon et al. Showed similar findings, not with the mefenoxam, but with finding Pythium aphanidermatum being very problematic and common on poinsettia. And so what are some of the possible explanations? Why do we tend to see aphanidermatum more than other species? This could be due potentially to the production season. So we grow poinsettia in the later parts of the year. So we have particular climatic factors that are at play. And it's also possible that the aphanidermatum has become established in some of the propagation areas, and that's why we tend to see aphanidermatum more common in the later stages and during propagation here in the United States. Forward, please. So one of the other things that Emma wanted to look at, and I didn't really want to talk too much more about her research, but she did take isolates from poinsettia and see if it could colonize other hosts that we found in the greenhouse. And so what was interesting is that she, even though she could find Pythium aphanidermatum on poinsettia, when she did her inoculation assays, she could actually infect petunia as well, but we didn't notice very many dramatic symptoms on petunia. However, we knew that it had colonized the petunia. And so this just highlights the point that asymptomatic hosts, so hosts that could harbor the fungus but not show symptoms, if they're in a production system, then it's possible you could, these could be reservoirs of disease, and that you could unknowingly ship infected plant material. So it's just something to highlight as a side note with some of this research. I'm going to see, so I probably, forward please. I will try to, okay. The next one I'll try to advance again. I tried just a few seconds ago, it didn't work. So one of the other things that we did is we looked at the cost to treat poinsettias with mefenoxam or with a product that we call Subdue Max. And part of the reason why we were interested in this is that growers still tend to use mefenoxam, even though we see some resistance. And so one of the reasons we feel why this is an issue is because it's one of the lesser of the expensive products in comparison to etradiazole. And so if you factor in the cost of labor as well as an application of 1 ounce per 100 gallons, you'll tend to see that, and these are with 6-inch pots, that one application across 10,000 pots equals roughly about $70. So that's pretty cost effective if you think about it. When you think about it on a larger scale, however, some of the larger producers in the United States, they produce, hundreds of acres of poinsettias. And when you think about it, that could equal $70,000 to $80,000 to treat every pot at least one time. So if you do have mefenoxam resistance, you have to realize that the cost of this application might be more costly than what you might think because of the efficacy, the lack in efficacy. So Emma's going to continue her PhD research. She currently works in the clinic at North Carolina State University. I've included her email address down at the bottom. We would like to offer this as a service through our clinic. And so if you are interested in looking into getting your Pythium populations in your greenhouse tested, not just on poinsettia, but on other crops as well, you should go ahead and email Emma. And in addition, she is really interested in looking at this pathogen in irrigation water. So she would like to collect some isolates and look to see how common Pythium, any species, are in irrigation water and in ebb and flood systems in greenhouses. And so with that, I'll try to advance the next slide, and we'll cover some other crops. So let's go ahead and focus on the stems. So I think I forwarded that myself. Let's go ahead and focus on some pathogens that attack the stems. So one of the main pathogens that's also very problematic on poinsettia, uh-oh, I don't know. Someone is forwarding my slides. Okay, it's not letting, okay, let me see. This is kind of frustrating. One of the pathosystems that attacks the stems is a very common pathogen in greenhouses as well, and it's Phytophthora. It can cause a crown and a stem rot. And so here you can actually see Phytophthora attacking the stem, and it produces this darkened water-soaked lesion along the line of the media, and it will cause collapse of the root system as well. And so it can be problematic. And the same story holds true with Phytophthora as well as Pythium. We tend to see resistance to Mefenoxam in some species of Phytophthora in comparison to other species. And so species identification can be really important of Phytophthora in greenhouses as well. And so if it is possible, if you are making large applications of Mefenoxam in your greenhouses, if it's possible to get a species ID, you might want to think about it, because there are some species like Nicotianae that have very mixed reactions, and then there are some species that are very sensitive. So I'm going to forward again. All right, so it worked. This is another pathogen that is problematic in the stems, and it's called Erwinia carotivora. But since then, some taxonomists have decided to call it Pectobacterium caritivorum. So you might see it listed in diagnoses either way. It does cause a bacterial soft rot. It does cause a bacterial soft rot, and you will tend to see the stems collapse. So this disease is typically the first disease that appears in a poinsettia crop, and the only other thing that it could be confused with could be Rhizoctonia cutting or stem rot. So let's go ahead and I'm going to forward the slides, and we're going to look to see what Rhizoctonia looks like. All right, so this is a picture of Rhizoctonia stem and cutting rot. And you can actually see the mycelium of Rhizoctonia. Rhizoctonia will often have an appearance that looks cobweb-like in structure, and as it gets older, the mycelium can turn reddish brown. It will cause a lesion right on the stem at the line at the top of the cube. And I'm going to forward my slide. It's not forwarding, so I've pressed it twice. Okay. Rhizoctonia stem and cutting rot can also be commonly found on leaves. I see. So I must have a really long delay, because it seems like it's fairly slow. I pressed it twice, and so I must have forwarded it. So all right. It's commonly found on, it's not really the slide I wanted. Brian, can you just go ahead and forward my slides for me, because it's not working, and I need the slides, I need them to go to backward. Okay, I like that one. That's a good one. So it's commonly found on leaves that become attached to the cubes, and they're typically infected under high temperatures as well as high moisture. And so these cubes hold a lot of moisture, and it can promote more of a foliar infection of Rhizoctonia. And it will allow the mycelium of the pathogen to cover the infected cuttings. Forward, please. It can also, you might not sometimes see the actual mycelium at the top, but you can still see the collapse of the stems. Forward, please. And if you were to look on the underside of that cube, you can actually see the Rhizoctonia getting started there. And again, it tends to look cobweb in appearance, and often as it ages will turn a reddish to brown color. Forward. All right, so the next couple of slides, I'm really just going to go over a few other diseases that are common, and it's not going to take very long. I think I only have about 5 more minutes. But let's go ahead and talk about botrytis. So botrytis is interesting because not only can it attack the stems, but it can also attack the petals or the bracts. And so here you can actually see botrytis stem rot, and you will see a canker. It can cause a crown rot as well as a stem canker. Sometimes you'll see the gray mycelium. We call botrytis sometimes gray mold. Sometimes you'll see the gray mycelium, but sometimes it disappears, and that's because the conditions that promote it are no longer present. Typically, botrytis can be controlled fairly decently with adequate ventilation and cooling and warming at night. And I'll talk a little bit about that later when we look at it when it attacks the bracts. Forward. The next slide that we're going to look at forward, please. So the next slide that we're going to look at is a picture of sclerotinia stem rot. Now, sclerotinia stem rot is not so common in greenhouses, but it does occur. It looks somewhat similar to botrytis. The one thing that's really interesting is that it will produce these very large sclerotia. So you can see on the top of the stem and a little bit to the left, you can see this circular structure. That is actually a hardened mass of mycelia. And this is how this organism overwinters. So if you have had sclerotinia stem rot problems before in your greenhouse, it's quite possible that those sclerotia are somehow surviving, and you might want to think about sanitizing the greenhouse. However, in comparison to Phytophthora and some of the other stem rots, this causes more of a dry stem rot, where you tend not to see water soaking. You'll actually see a discoloration, and you'll see this gray fuzzy growth. And as the infection occurs, you can also see these sclerotia that are forming on the stem as well as in the stem. Next slide, please. All right, so I'd really like to talk a little bit, and it's one of the last diseases that we're going to go over that is new in this webinar. It's scab. And the reason why I want to talk about it is that I find it really fascinating because there aren't very many diseases that can actually cause what looks like a growth effect. And so it really causes a gibberellin-like effect, and it will promote the excessive growth twice as long as other branches. So if you look really closely at the picture on the right, you can actually see these elongated lesions on the stem of the plant. Next slide, please. So scab is actually caused by a fungus, and it's the fungus name is Phacelloma poinsettiae. It's not very common in greenhouse-grown poinsettias. It's found more commonly outdoors in outdoor poinsettia production, and we typically will see it in Florida, Hawaii, Puerto Rico, and other areas that they do outside production of poinsettias. However, what's interesting about it is if it's detected early through normal roguing and treatment, it can be eliminated fairly quickly. And so if you have very abnormal growth, abnormal elongation, you might want to look very closely at the foliage as well as the stems to see if you can't see any of these scabs. Forward, please. So this is what it looks like on stems. And essentially on stems, it will produce scab-like raised lesions. These lesions will often have tan centers, and these lesions also have a color that surrounds the lesions. Sometimes it'll be white, sometimes it'll look red, and sometimes they'll have purple margins. And often you will see a number of these lesions as the disease gets established, a number of these lesions being produced on the stems. You tend to see this occur first. Sometimes you can see it on the leaves as well. Forward, please. So again, here's some pictures. And again, you'll see multiple lesions on these stems. Sometimes they're circular, sometimes they're elongated, and they're scabby, so they're usually found on the surface. And again, they can be round or elongated, and they usually have a tanner center than the outside. Next slide. So let's go ahead and talk about overall. Let's talk about overall diseases as well. And this is going to mainly be botrytis as well as Xanthomonas and powdery mildew. So botrytis blight. Botrytis blight is again, it's problematic. I showed pictures of it, of what it actually looks like on the stems, but it can attack any above ground portion of the plant. And you tend to see a sporulation of the growth. It can look gray to off-white. Sometimes you'll see a tannish color, and again, it produces very large amounts of sporulation on the surface. So this is what botrytis blight looks like. Forward, please. And what I mentioned about botrytis, and I'll mention it again, is that it can be fairly well managed with venting the greenhouse and with heating at dusk. And basically you want to do this to lower the overnight condensation on the leaves as well as the bracts. Another problem that is emerging or is becoming more problematic in poinsettia production is a bacterial blight. And bacterial blight is caused by a Xanthomonas species. It's caused by a species, Anoxapodes. It was first found in the 1950s in India, and it showed up in Florida in 1962. And many of the popular cultivars can be quite susceptible to this disease. So one of the first things that you notice, forward please, is that the spots are first visible on the underside of the leaf, and they'll often look gray to brown. They will produce water-soaked lesions, and as these lesions enlarge to 2 to 3 millimeters, they can then become visible on the top side. And typically you tend to see numerous lesions throughout an infected leaf, and these lesions can coalesce, they can grow together, and they can cause a larger blighting on the entire bract or on the entire foliage. So the spots are chocolate brown to rust colored, and they can be surrounded by a pale green to yellow halo, depending on the time of infection. And occasionally you will see distortion in heavily infected leaves. Next slide, please. So I'm going to show you what scab looks like. We talked about scab, about what it looks like on the stems. This is what it looks like on the foliage. It can be confused with Xanthomonas blight as well. So this is an example of when you might want to get an accurate diagnosis, because these are controlled completely differently. Bacteria tend to be controlled with formulations of copper as well as clean grow, and scab really needs to be robed in order to eliminate it. Forward, please. This is another picture of what scab looks like. Again, you tend to see they look like raised blisters on the foliage. Forward. So this is just an example of the raised lesions, again, of what scab can do. Forward, please. And this is what powdery mildew looks like on leaves. You tend to see it on the top side as well as the off side, the bottom side. It looks like powdered sugar often on the foliage, and if you turn the leaf over, you tend to see a white sporulation. The beginning stages, you will see a yellowing, a discoloration as well. Forward, please. All right, so I'm just going to show you a few more pictures. We've really talked about the main diseases, so we can scroll through these fairly quickly, Brian. Let's go ahead and forward. Botrytis can also cause spotting on bracts, and so here you can see what it does on petals. They'll start out as tiny flecks of discoloration and can cause some distortion as well. And so Botrytis blight at this period of time is very problematic because it does affect the aesthetics of the plant, and this is when Botrytis needs to be heavily managed through proper ventilation and warming at night. Forward. And I don't really need to explain any more of the slides, so we can just forward. This is a Botrytis blight again. It can look like a desiccation on the outer sides of the bracts. This is a heavy infection of Botrytis blight. Forward. And this is powdery mildew. It looks quite different on poinsettia than Botrytis as it gets older in age. And again, it looks like powdered sugar. It looks like you've dropped powdered sugar on the top sides of the leaves. So I think that is, oh, there's one more picture actually. There's a picture of Xanthomonas blight. Xanthomonas, or bacterial blight, will cause more of a smaller lesion on the bracts. They look water-soaked. And again, you can see them on the top side as well as the underside of the bract. So with that, I'm happy to take any questions. I think we have a few minutes for questions. And I'm assuming that Brian Krug will go ahead and answer what questions, if there are any.
Yeah, so if anybody has any questions, please just go ahead and type those in to the question/chat portion of the toolbar that you have for GoToWebinar. So far, we don't have any questions. All right, just to keep things moving along and on time, we'll take a short break here before we go on to Brian Whipker. So it is 1:49. So at 1:54, we will start again. This concludes part 1 of the 3-part series of Poinsettia Diagnostics webinars. Please view part 2, where Brian Whipker from North Carolina State University will cover common nutrient disorders of poinsettias, or part 3, where Raymond Cloyd will cover insects and mites of poinsettias.